Bismillahirrahmanirrahim. Transcription is the synthesis of RNA from DNA template. It is carried out by RNA polymerases. RNA polymerase catalyze the formation of phosphodiester bond between the nucleotide that base pair with complementary nucleotides on DNA template strand. But unlike DNA polymerase, these do not require primer for initiation of RNA synthesis. A strand of DNA that serves as template for DNA synthesis is copied in the 3' to 5' direction. Synthesis of new RNA molecule occurs in the 5 to 3' direction. The ribonucleotide triphosphates ATP, GTP, CTP and UTP serve as precursors. Each nucleotide base sequentially base pairs with complementary deoxyribonucleotide base on DNA template strand. Adenine base pairs with thymine on template strand. Guanine base pairs with cytosine on template strand. Cytosine pairs with guanine on template, whereas uracil joins adenine on template. The RNA polymerase forms an ester bond between the alpha phosphate joined to 5' hydroxyl group of the ribonucleotide, which is destined to base pair with complementary base on DNA, and the 3' hydroxyl at the end of the growing RNA chain. The cleavage of the high energy phosphate bond in the nucleotide triphosphate and release of pyrophosphate from the beta and gamma phosphates provide energy for this polymerization reaction. Subsequent cleavage of pyrophosphate by pyrophosphatase helps to drive the polymerization reaction forward by removing a product. So that's the pyrophosphate that has been released, and that's the bond formation, phosphodiester bond formation. And plus the hydrogen bonding between the guanine shown by pink color and the cytosine shown by magenta color here. The double-stranded DNA consists of a coding strand and a template strand. The template strand is the strand that is used by RNA polymerase during the process of transcription. It is complementary and anti-parallel not only to the RNA which is being synthesized but also to the coding or non-template strand of the DNA. The newly synthesized RNA is complementary to the template strand but not identical to it. In contrast, it is identical to the non-template strand with exception of uracil in place of thymine. So that is why the non-template strand is called the coding strand or the sense strand or the plus strand. And it is this strand on which genes are present. Contrary to it, the template strand is called the non-coding strand or the antisense strand or the minus strand. During the protein synthesis or translation, messenger RNA is read 3 to 5' in sets of 3 bases called codons. These determine the amino acid sequence of the protein. Thus, the base sequence of the coding strand of the DNA can be used to determine the amino acid sequence of the protein. For this reason, when genes sequences are given, they refer to the coding strand. The base in the coding strand of the gene serving as the start point for transcription is numbered +1. This nucleotide corresponds to the first nucleotide incorporated into RNA at the 5' end of the transcript. Subsequent nucleotides are numbered +2, +3 and so on. Untranscribed sequences to the left of the start point, known as the 5' flanking region of the gene, are numbered -1, -2, -3 and so on. By comparison to a river, the sequences to the left of the transcription start site are said to be upstream for the start point, and those to the right are said to be downstream. For the genes to be expressed, RNA polymerase must recognize the appropriate point at which to start transcription. The signals or the DNA sequences that RNA polymerase recognizes as the start sites for transcription are called promoters. Promoters are short sequences in DNA. Promoters are short sequences in DNA called boxes or elements. These determine the start point and the frequency of transcription. Because they are on the same DNA molecule and near the gene they regulate, they are called cis-acting. Certain consensus sequences in the promoter regions of gene to be transcribed guide the binding of the RNA polymerase. A consensus sequence is a sequence that is most commonly found in a given region when many genes are examined. In prokaryotes, an adenine and thymine rich consensus sequence in the promoter determines the start point of transcription. In E. coli, this sequence is TAT-AAT, also called TATA box or Pribno box. It is located at -10 and recognized by sigma factor. Bacterial promoters contain a sequence TTGACA in the -35 region. In bacteria, the binding of RNA polymerase with sigma factor to promoter region of DNA causes the two DNA strands to unwind and separate. Within a region, approximately 10 to 20 nucleotides. Initially, the RNA polymerase docks at promoter and begins to synthesize a transcript of DNA sequence. Several short pieces of RNA are made and discarded. Elongation phase begins when the transcript, typically starting with a purine, exceeds 10 nucleotides in length. Sigma is then released and RNA polymerase is now able to leave or clear the promoter. Now it starts moving along the template strand in a processive manner, serving as its own sliding clamp. As the polymerase transcribes DNA, the untranscribed region of the helix continues to separate, whereas the region that has been transcribed rejoins its DNA partner strand. Unwinding generates supercoils in the DNA that can be relieved by DNA topoisomerase, which have strand cutting and strand resealing abilities. Topoisomerase 1 makes a transient nick in one strand of the DNA. Intact strand is passed through the break before it is resealed. Topoisomerase 2 works by making transient nick in both strands and relieves supercoil and reseal or act by introducing negative supercoils in prokaryotes. In contrast to DNA polymerase, RNA polymerase does not have the 3 to 5' exonuclease domain for proofreading. If a wrong ribonucleotide is inserted, it causes the RNA polymerase to pause, backtrack, and cleave the transcript and restart from scratch. Nonetheless, transcription has a much higher error rate compared to replication. The elongation of single-stranded RNA continues until termination signal is reached. Termination can be intrinsic when it occurs without participation of other proteins or dependent upon participation of an additional protein known as growth factor. So, here you can see that when it reaches the termination sequence, it is paused. RNA polymerase is paused. Now coming to row independent termination. This type of termination is seen with most prokaryotic genes. This requires that a sequence in the DNA template generates a sequence in the nascent RNA that is self-complementary. This allows RNA to fold back on itself, forming a GC-rich stem plus a loop. Additionally, just beyond the hairpin, the RNA transcript contains a string of U's at the 3' end. The bonding of these U's to the complementary DNA strand is weak because of the presence of two hydrogen bonds between adenine and uracil, in contrast to three hydrogen bonds that join cysteine and that join cytosine and guanine. This facilitates separation of the newly synthesized RNA from the DNA template, and the double helix of DNA zips up behind the RNA polymerase. In row-dependent termination, row protein moves along the RNA until it reaches RNA polymerase paused at the termination site. The ATP-dependent helicase activity of the row separates the RNA-DNA hybrid, causing the release of the RNA.